officially welcome you to our seminar session.
Yeah, thank you.
Dr. Dieter, thank you so much for your time and availability. Can I just briefly introduce you to the participants?
Yeah, please.
Before we start. So Dr. Dieter has a background in mathematics and computer science, as well as in crop science. He has developed software systems for wheat breeding initially, and then during his PhD in plant breeding and quantitative genetics, and he also developed systems for that are used for many breeding programs. He has background in studying the genetic architecture of quantitative traits, mostly in bred wheat. And now he had a postdoc in Syria and then joined the University of Nebraska, now as an associate professor in cereal breeding. In the 90s, he founded Agronomix Software and has trained many breeders to use AGROBASE. And more recently, looks forward to the global adoption of Genovix, which is the new plant breeding software that he's going to present to us right now. So thank you again for your time, and it's a pleasure, big pleasure for us to have you here.
Okay, thank you. Yeah, so I'll start with this PowerPoint presentation. So we started in 1990, and we have a global client base, including universities, government, research organizations, and we're proud to have 3 PhD plant breeders on the support team. So shall I say that plant breeding is in the company DNA. It's a system for variety testing, plant breeding, and it's a comprehensive one. We've worked with many breeders of many crops, whether it's annual, biannual, perennial, hybrid, vegetable, horticulture, and a lot of field crops. You'll be seeing a lot of experimental designs in the variety testing system. There's a breeding system for many crops and the cropping systems, seed inventory module, and we have a lot of integrated analytics, both our own code and the use of open source R. And there's a Genovix mobile to capture data from Android, iOS, Windows, and I imagine in Brazil, probably Android is the most popular. It's a SQL Server backend database with a .NET interface, which you'll see. And it's a highly relational system, which means that everything is keyed. You change one thing, it ripples through the whole system, and you can pull data and do things that is not possible in most other systems. It's multilingual, including recently Portuguese. And we have a new bioinformatics module. So here's the interface. It looks kind of like Excel, and with the ribbon bars. And here I have an experiment open. It 1 click brings us all up. And this shows here the traits. Trait names, of course, can be customized to the crop you're working with. So if it's flowers or sorghum or sugarcane, that'll be different from the wheat breeding kind of traits that I have here. And a lot of icons and tools to help the user really move fairly quickly through the whole thing. And as I said, it's also in Portuguese, as you can see, from our translator has done, from what I can tell, a fantastic job. And what I haven't translated is the name of all these columns here, which there are systems options to change those, as well as the trait names, put them in Portuguese, build your data dictionary, and you're good to go in that language, sort of facilitate the use. For larger organizations, especially universities that have a lot of students and they want to get a student license, it's really helpful to be able to control what database a researcher or student may enter, and also once they're in there, what they can do, the whole thing of permissions. So there are a lot of right-click tools to make it very, very efficient. So with my 30 years experience in our team working with experiments and researchers, we know that sometimes you need to find an experiment if you have many. Maybe you need to modify the genotypes in it and so forth. So there are a lot of right-click tools that are very practical and have been developed over time. Same thing for genotypes or germplasm. A lot of tools here as well to organize all the genotypes. So you could have easily 100,000 genotypes, put them in different groups, organize them quite efficiently and quickly, which not as easily done in Excel at all. There are a lot of filtering tools. Here's a very simple one, protein maturity, powdery mildew, and something about a name. And you see here, we end up with 68 records. So all those filters can be saved and used over and over again. Again, it speeds up the use of the software. And if you're in hybrid breeding, it's important to pull up information, even self-pollinated crops. Sometimes it's nice to know where certain germplasm has been used as a female parent or a male parent, or just does the female pedigree contain something. And then from here, we see here AC baton, or just the letters AC, found all in here. You can create a group from there, and then probably in minutes do an SQL query and pull out all the data from what you found. Again, accessibility is important to assist research. And with such an integrated database, we have the ability to have dynamic SQL instant queries. So Genovix will build them on the fly. You can save them, use them over and over again. So here I've said, of these genotypes, I want to have these 4 here. I want all their data. And I chose a number of locations, number of years, some seasons, experiments, maybe some nurseries, some traits, and then click. And really it's only seconds, whether it's 5,000 or 50,000 in a search, it'll pull it all up. And here we see grain yield. I've just clicked name one, name of the first variety here, and scroll down, you see them all. So it pulls up the data. Of course, GPS is important. You sometimes experiments are not on the official research station, but in a farmer's field somewhere. But even as at a research station, it's good to have the GPS coordinates here. So this is Brandon, where we have clients using our current system. And there's a column manager. So this has evolved over years. We found a lot of researchers spend quite a bit of time organizing columns, group of columns. So we have a universal column manager. So on a list of data traits and system traits, you can decide which ones go into a given view. So here you see for ADYT, Advanced Durum Yield Trial, BR for Brandon. We have all these potential views here. Some more helpful than others. And anyways, you just click, assign it as the active view. And just by changing the click to another view, you have instantly changed the data, whether it's one experiment or 100 experiments that are open at the same time. So flexible trait names, again, I've mentioned, because if the sugarcane and potatoes and wheat and sorghum, very different traits. And maybe you want to say like oil content percent, ploidy level, anthocyanin color. So you can name it what it is. And then actually you could, even when it's assigned in the database, I can say I want oil content, PERCENT, one click, and it's changed to the entire database. It could be in 500 experiments, click, instantly done. So that's because everything is linked and interrelational. The locations, it can be for research agronomy. This is, I think, important. That's why it's in here, is the ability to add things like altitude, soil. So these are traits for the locations. So if you're ever want to do a meta-analysis where you have location traits like a soil type, and then you might have other factors, you can add more traits and link that in the database. everywhere that location is used. So years are linked automatically. So if I create trials for 2020, then instantly that node here is added, and all the experiments appear under it as per location. So it's all very nice and hierarchical. Now, experiment nursery traits. We all know that in agronomic research, if the previous crop was wheat, there might well be a different response than it was if it was beans or soybeans, nitrogen fixing crops. Wheat and maize tend to deplete nitrogen. So you can add that as a trait. The planting date, the technician responsible, all these things. So again, experiment management, but these could be part of a meta-analysis. So you have location traits, experiment traits, the actual plot traits, and link them into one large file, and consider other analyses like ridge regression. So relational database here, we see how you can link the genotypes, location, seed lot, experiment. So what I've done here is plot traits is what you normally take. So in the data, so here I've got grain yield, powdery mildew, protein, maturity. And you see here, blue means that it's, the trait is beyond the limit that's typically expected. So it might be an outlier, might not be. Over here, we see one that's red. Maybe it's being less than the minimum value of 1. Is it a suspect value? So we got the plot traits, and I click on genotype traits. So the actual genotypes in the experiment, they can have any number of genetic traits here. So even if you had like 50 SNP columns or marker or whatever, you can click and add that all in here. And they're the normal traits, the usual name, but superseded. In my case, I like a asterisk in front for simplicity and readability. And then I've got location traits. I just use an ampersand. You can see that's the location attribute. And I've got a seed lot. If we have a seed lot system, this is where the seed came from. The seed inventory, the germination percent, I just put it in brackets. And then the experiment trait, I proceed with the ampersand or the @ sign. So plot size 11. So the thing is, this can be all in one view. Create the view, it's all automatically linked. So you decide which traits, link it, and it works right directly for you. So we think that it really extends the capabilities of research. And the thing is then that, again, if you want to do a meta-analysis, this easily ties the data together. So many functions for calculations. There are about, I think, 200 or so, numeric and character traits and date traits. So no need to resort to Excel. It all works right in the system. Subsamples. This could be for individual plants. So in a nursery, maybe you're taking, let's say here 6 plants, and I want to take in a population 6 scores for a certain trait. Or I'm doing 6 readings for disease progression. Maybe I'm looking at powder mildew from the first date to date number 6. What's the progression of the disease? So that could be then the 6 readings. So it's either repeated measures or traits. And then even there, you can calculate the average, the skew, kurtosis, or whatever as a computed value. And again, that could be many thousands of records done instantly. Everybody asks, well, can I export to Excel? Yes, 1 click and it's to Excel. That's never a problem, or a PDF or text file or CSV. Images are important. Some crops, it's really important to look at the image of the genotype, maybe the seed, the fruit. Another one could be the disease. So what does susceptible in one plant look like as susceptible in another one? So many different reasons. And we found that even field crops breeders tend to want to use images now. Initially, this was really just for vegetable crop breeding. You could even go further and say, well, click on this icon. There was an icon, this one here, I think. And it shows you the image of the parents. And then if you took many photos, you could scroll within it and see how it all works, as well as the data of the parents and that genotype. So many people have their data in Excel. How do you get it in the system and then linked and organized? Well, I won't go into much more detail other than we can import experiments, nurseries, genotypes. We have people who have imported data from 20 generations back, 20 years of research data. And we've been able to link all the filial generations, let's say from an F8 all the way to an F1 and then the parents. And that builds a nice dendrogram as well. So that's pretty important for the history of a trait. There may be a lot of lab data that comes in, so you want to link everything like balances and other devices. And then so you can set it all up with the port name, the bits per second, and all that good stuff. And then you can import it directly, scan a sample into the database. Reports, maybe it's kind of germane, but is still quite important. Here, this is just, I think, 3 clicks, and I generated what we call a quick report from anything in the system. The report generator, here, I will simply say you can see a lot of icons to do things like barcoding report. We can do all kinds of graphics. We can do sorting, grouping, bring up the field list, link other tables that are in the database. It's quite a powerful system. And we're still figuring out how to use it all actually at Agronomix. But it's a great system that we have. And labels, pretty, commonplace, but here's just quick code. You can easily design those. Again, a lot of material. If you're doing agronomic or breeding research, you need to print labels quickly and accurately. So variety testing, this is the engine that runs the application. Many experimental designs with analyses built in or via open source R. So we can go with trials across locations, years, and seasons, and G by E analyses integrated with breeding and so forth. So later, in the live presentation, I'm going to look at the different one experimental design. I think I'll do an alpha, but you can see if you're doing just agronomic research, you want to do a split plot or a split plot or a strip plot or a factorial. Those are possible, as well as non-replicated designs. And now it's showing here is the augmented RCB design that we have added recently. So it's easy to look up and see where all the experiments are for a genotype. So here I just click on ADYT and I bring them all up like so. And then here I'm actually creating an experiment. So I'm going to use these entries here, put those in, and I wanted only 28 for a design. And that's the interface, and we'll go more into that momentarily. Perennial experiments. There are some crops where, like tree breeding, where you plant it and it's going to grow for many years, maybe 7 years before you see any fruit at all or longer. And anyways, this system allows you just click and add years, seasons as well. So here I've got 3 years and 3 seasons, and I've got subsamples as well. So you see here the season, somewhere here has got the year factorial, the location. So 2020 to 21, and then we got the 3 seasons. So there's some crops you have to structure the database that way. That's the only meaningful thing, really. All right. So for statistical analyses, we have inbuilt RCBD and other designs, moving means, nearest neighbors, or analyses by R scripts, the matter of just using it here. So here is a spatial analysis, nearest neighbors, in which I have actually co-authored a number of papers over the years. And you can see these are the estimated trends. This is my experiment where this was dry patch of soil, wet, dry, wet. It was actually uneven sprinkler irrigation, and it was a bit of a disaster. But fortunately, I learned about nearest neighbors analysis, and I eventually wrote all the code for it. So here, for example, we see variety that under RCBD regular analysis you think is number 5, and actually is the best one. It ranks number 1. And here's one here, 21st. No, it's actually 10th. You probably wouldn't select it if you're getting the top 5. But the breeder wants to have the best genetic ranking of the entries and nothing else. That's really what counts at the end of the day. We can analyze by R scripts. So here I've got the Agricoli package, and I install mass and then NLME if it's somewhat unbalanced. And here we see the unadjusted and the adjusted and so forth. And yeah, so that's how we integrate the use of R, which runs externally, and we bring the results back in the system. Once the analyses are done, the experiment means are stored in the system. You see right here, as well as all the stats. And down here, one could click and bring up the analysis that was responsible for those means, which is a bit of an audit trail. It's always good. And for G by E, we have ggbiplot, and this uses the R package for ggbiplot. I believe it came from University of Sao Paulo, if I remember the reference correctly. I might be wrong, but I think that's where it came from, a contribution to the R community. And Shukla analysis, so for multi-location trials, this is one way, again, of looking at statistics, equivalence and R squares and all those good things about interactions. And again, more statistics on looking at adaptability. And numbers are great, but a picture is worth 1,000 words, I think is the Chinese say. So here we see that Grandin is really doing really well. BRS 80, not so well. I think it's in here somewhere, but shows you the interaction of the different varieties across the different experiments. And this is the average of the experiment for that trait. Now, hybrid breeding, hybrid crops in particular, do a lot of what we call head-to-head comparisons. So here we go through the whole database with a structured dynamic SQL query. We find 45 experiments where these two varieties are in common. This is the controller to check, AC Baton. And we see here that in 2020, it's significantly different, actually better, as opposed to 2019. And it gives a breakdown of each location, whether its statistic is possible or not. So again, once the data is in the system, it's just a few clicks to do this sort of thing. And it goes across years. We can look at the usual heat map. A lot of researchers want to look at for their data, but I think it's better to look at a 3D surface. So you can easily go in our system, a few clicks, and look at the 3D surface. And that experiment that I showed you earlier, this is the dry zone, that's the wet zone. And furthermore, if you do the biplots, this is a good exercise before you analyze an experiment to take a look and find out, you know, what really is happening here. What are the assumptions? And here, classical ANOVA probably doesn't apply. So here's a contour plot. I think that's a lot better to look at the data. And once the experiments are designed, of course, field plans are quite important. So we can just line them all up and prepare for planting all the different experiments. Germplasm management. So we have a link to multivariate clustering in R. So this one here uses some marker traits as well as some phenotypic, and there are options to standardize them and use the, I think it's the Manhattan clustering algorithm. There are 7 available through R. And then we can see that these varieties here as a group cluster differently from those. And that might be indication for what we do for breeding. So here we have the dissimilarity matrix. And the other thing is we can add, while we're looking at U-types, not only images, but documents. So sometimes that's important for If you're doing even public research, maybe there might be some genotypes, transgenic, non-transgenic, that sort of stuff. It's important to know that, or whether a gene has been patented. So instant view of all genotypes and their data. So here I'm looking at ADYT, AC Baton, and one click. And on my laptop, it takes about 6 seconds, and I pull up about 12,000 records. It's all the data for that genotype. Same you can do for traits. And I can rename a variety as we see here. So for germplasm development, we see here, this is cytoplasmic male sterility. We handled that as well. And for each genotype, I could show the history. How was it developed? It's all actually nicely linked together. Weighted selection index. I talked about filter, but there are a lot of papers that have been written on selection index. So we can assign weights in Genovix, truncation values. This would be an example of truncation selection with a weighted index. And that results in these values here you see in the blue. It tells you the genotypes that win according to this index. So again, for the breeder, research, another tool. Again, AC Baton is in 163 experiments, showing again how things are linked, and it makes it just easier to work with. The system, saves time. Breeding module, we can import genotypes. We can compute a nice dendrogram. You see here all the pictures individually, and then I've got the data. So if I have molecular markers, I want to make sure that those alleles come down the generations as I expect. How does that translate into the phenotypic trait? That's the big question. So I can pull the data up cross generations, look at the photos if I want, and make my inferences. So the breeding method manager can be customized for any crop, every crop. So what you will call the naming rules, again, everybody has to do that in Excel. Why not have the system do it automatically for you? And cross prediction. So I've got two marker traits here. We could have a genomic prediction value, which we'll get into in a few minutes. And then I can set this up. I can have and or and all kinds of complex rules. And then we see here that results in the genotypes. The combinations in green are the ones that win, that you could consider. And we have a number of users who just spend quite a bit of time going back and forth, adjusting the criteria set that we call it, and helping decide what they will actually put in the crossing program. It's just a tool in addition to what data science may give you. And then these can be saved and confirm the crosses and go into the system. Once in the breeding system, we can open up a crossing block, be created. So there are quite a few different breeding methods that are available. And from there, you can generate the next set of genotypes, F1s or whatever. Population advance. So here I've selected plant number 5, 8 plants at plant 7 to 12 and so forth. Again, working with breeders for about 30 years, all these small things just come up that they need that flexibility. And everything in Genovix goes into a group somewhere, a folder, so it's well organized and tight. So in this case, the naming rule is just take the name of the population, add the plot number, and what we call the advanced code. So here we see this is the population. It's plot number 6 over here. We see 6 down here. And this was the one where we went 7 to 12, if you remember. Then we had letters A to G. So again, quite a bit of flexibility. Now, in hybrid crops and getting hybrid seed production, you might want to have females, and H here is the F1 and M is the male. So again, this system allows for many different combinations and possibilities, laying it out in the field, whether it's an isolation nursery or you're simply making more hybrid seed. Maybe you just want 3 rows of F, 3 rows of the female, 1 of the male to pollinate. Again, whatever is required by the breeder can be set up. Parental data is easily linked. I have worked as a breeder, and I want to know I'm getting response to selection And this also may help decide what plants I will, what population I will select. So here I'm going to look at the leaf resistance and the marker information of the male and female parents. And we see here the marker of the female right here, and then the marker of molecular marker 1 in the male, missing here, I guess. We don't have that data. Powdery mildew of the female and of the male over here. So all that data is there in the gray. It's only changed in the genotype table. And here in the gray, and here in the green is the data that's coming in this particular nursery, it's F3 Advanced Nursery. So again, because it's a relational system, we can make those linkages instantly. We also do parallel line by tester analyses, although there are other sophisticated models available. It gives you the GCA and get the SCA as well. Inventory module, I will just mention that we have integrated a whole ability to connect seed in all parts of the system and to do, so whenever I look at a genotype here, I can look at the seed source, the seed allocations. I know everywhere common it's gone. And here I have the inventory allocate to experiments. So I clicked on CPYT BRKV, click here on seeds, and I think it was 20 grams per rep. So by 4, it's 80 grams allocated, quantity available. This is where it comes from. Again, not highly scientific, but in a practical breeding program, you need to keep track of that, and packing list and so forth. And I can link the genotypic traits with the traits in seed inventory, and those are all customizable traits, whatever is relevant. Genovix Mobile, I can go with in my field or greenhouse with Android. A lot of settings here. Click and enter the data, and then there it's uploaded to the database over the cloud. Genomic prediction within Genovix. Why? No need to export and change data formats. All workflow for making crosses, applying nurseries, data collection on the population in one software. So all links together. The analysis done in a few clicks, predictive values directly integrated back into Genovix. So here's an example. The data is in Genovix here, a couple of options which are relevant. So we do a lot of pre-processing that normally people spend a lot of hours with their SNP data and everything, getting it all organized. No need to do that. There's a lot of settings here that our two people have written this, our experience in breeding and bioinformatics, and they have a lot of the relevant tools in there and options. So it's parallel computing. Some of these may take one run we did had, I think, 250,000 SNP columns, able to process it and link it all back right into Genovix. Didn't take very long. And then it's easily integrated back in the system. So the other consultations available at our company from this kind of expertise. And here we go with the cloud computing and that. And we got tutorials. So now I will look at the software itself. I think I've got about, I believe, about 15 minutes left, something like that. Okay, so first of all, I'll open up CPYT BMV. You can see here, one click and system opens up. So here's the details, all the data validation tools and that sort of thing. And the analysis we talked about is easily done. So here's the alpha, and I click grain yield like so, and away I go. And the R analysis done the same way. So, and again, the linked images. So you see here, you just click on the icon and that control. This is an image for the experiment. I've got one here now for the variety itself. And I go here to traits, and again, traits all decided and organized by the user and so forth. With The R analysis, a lot of people wonder, like, how can I integrate that? Well, we provide the structure to set things up so the data comes out in a certain format, and then you could write your script in here and add that in the system. It's available then in the lookup list for the analysis of the traits. So that's kind of how that works. And so we try to make it open-ended. And the script that we just saw, the one is lattice test. That was actually on the PowerPoint. This is the beginning part of it. Has kind of the same structure, and then later on comes the model. So as well as using a lot of R, we allow the ability to leave the user to leverage even more with their models that they may want to put in. So I'll go on and I'll quickly design experiment, actually a trial. So I'll go here and it's say, okay, I'm going to say USP 102. And I'll enable a trial. And I'll go down to alpha design. And I'll click the name in here, say USP 2021 B, because I think I did one earlier, so I'll call it B. Then I'll go to design parameters. So say 28. Oh, back on the experimental designs. So we have split plot, split plot. It's a lot of factorial designs for agronomic research, non-wrap designs for early generation testing. Lattice square, this is the two-directional resolvable lattice design in the augmented randomized complete blocks. So quite a number of designs to cover different needs. So again, we go back to the alpha design, 28, say 2 complete blocks, 2 entries per block. This would be a 2x14. Should have relatively good precision. Locations, I'll go to Brazil, and genotypes. Let's say I'll choose these here. I have more than I need. I'll just take the first 28, pop it like that. And then I go to traits. I could say, well, I'll certainly start with grain yield, but I can add more traits later. Then I go to groups, and I have a group already called USP right here that I just made earlier today. And for the trial, again, things are organized in Genovix. Click on summary, use location code. So that looks fine, differentiation everywhere. Click preview and say, yep, that looks about the way that I want. And again, these steps are the same for all experimental designs. Then you click create, and it tells you it's created. Now I'll do a right click, and I'll just go down to USP right here. So here are the 102s. So that's one I just created. So really, it's fairly efficient that way. And maybe I just show one thing here is the crossing matrix tool that I think has generated a lot of it. Again, we could have biparental crosses, and we're just finishing polycrosses at the moment for crosses that, crops that use polycross. So here's biparental. I click here in female. Maybe I will use the 80YT entries like so, and male parents. Again, I could do a diallel of them, but I'm just going to use this group here. So the CDC varieties use the crossing matrix. So I think this is about, 377, 130 out of 377. So there we go. And then we go to potential crosses. And again, we could save those in a group and go on. So in fact, I'll just go here and say it's going to be in this group here. Click create. So I have the naming rules set up, and then I can click create it. It's in the database. So time does not permit me to go much further than that. So close here. And under breeding system, maybe I'll just briefly look at the popular. Yeah, we'll look at the series 1 crossing here. So again, all the different crossing tools. So I go to breeding and I say, maybe I want to cross this genotype, Cody, with Arapaho, and I choose the method. So you see that it's all consistent, the system, how it works. Regular cross here. Then I could go and say, well, you know, I have predicted crosses or preferred potential crosses. So I'm going to choose those. Again, it's regular cross, meaning there's no CMS stuff involved. Create the cross in genotype. All females with all male plots, and I think it's series 1, 2021 crosses. And I click like so, found them, 46 crosses that were planned. I click here and against the database. So I think the message here is that it just will save a lot of time to use this system to work with it and adapt it to the different crops. That's pretty well what the message is. And then on the breeding system manager, I go down here, here's a regular cross. And again, I'm going to say USP starting at the number 1. Close that here. Close that there. And choose those, and I can go ahead and generate. And the system checks everything all the time. Something like Excel, you have to kind of watch it yourself. But here, because it's a linked database, not only is everything well connected, it checks all the time. So here, well, I'm missing the breeding methods. Can't go ahead. Okay. One last thing, and I'll finish. I'll just cycle back for a second to experiments. And I was going to say that under perennial experiments right here, you can see that we've got the different years. So under details, I got the 4 years and the different seasons. And as we scroll down, we'll see it's all there. In this case, 864 records. So again, that's flexibility for the different crops that are involved. You know, in different research programs, even different breeding programs. There are some crops where material is harvested for a number of years, and then selection is based upon the GCA combination of the parents involved and so forth. So yeah, and I'll just mention here the seed inventory you saw. So everything is linked there, and we can bring up seed lot groups and so forth. So again, it's depending on the research you're doing, it may be quite important to have control of the seed that's being used. So I think that's pretty well it. I've only shown maybe about 5% of the system, but anyways, I'll leave it open to questions. I think that's kind of the time we agreed on.
Yes, thank you very much for your presentation.
Yeah.
I think it's very interesting for students to see perhaps a different reality that they live now and that they're gonna face in the near future.
Yes.
So it's a good preparation for them to have an overview of possible work environments for them. Let me see if I have some questions. So we have, let me check, 15 minutes for questions.
Mm-hmm. Or 17 if you want. I'm here.
Yep. Questions, anyone?
Hi, I have a question, Gabriel and Dieter. First of all, I would like to thank you for this opportunity. In fact, I have some questions, but I will focus on 2 of them, right? First is about the possibility of integrating imaging data by drones as genotyper for selection. I ask this because I saw days ago about the news about the partner with Progeny Company from Purdue University. And another question is, do you have a special plans for university to use this system? Thanks.
Tell me again your first question. I didn't quite. Are you talking about data from drones?
Yeah. This is the question.
Oh yeah. We have a partnership with Progeny Drone, so we can already link some of their image data. They have what's called plot clips. So that isn't fully functioning yet, but we will have the ability. We already have the ability now to open up an entire field plan like here and see that the drone. I know the people very well. The drone will fly over an entire field. You got the GPS coordinates. So you export this plan to the drone software. See, here's the grid that has somewhere I've got the range and row numbers here, plot range, plot column. That maps the position, okay? So the drone flies, they tell it the GPS coordinates of where it begins and ends. Then they do the algorithm to run everything. And when it comes back, you can import this whole sheet here for all the experiments involved. And you would have the final data, right, that comes from the algorithm. Maybe it's emergence, stand count, and that sort of thing. The importing of the images is a little more challenging because you have to have a known naming sequence. The system says, well, image is great, but where do they go in the system? So we've already agreed that there is a unique naming they'll have, which is a combination of the plot range and the plot column. So that's the answer there. At the moment, there are larger files that they have of the whole field, which we've imported successfully. That's not a big issue. And number two, for universities, yeah, we have one American university that's going to license this to students because faculty members already are using the system. And AGROBASE, we have had long-term license with the University of South Africa, Bloemfontein, and there's a number of other universities where faculty have been using Genovix to actually teach to use it in their plant breeding course. And that's one of the reasons why I have to date 115 Genovix tutorials. So students can watch the tutorials, learn the system, as well as what their professors teach in practical breeding. So does that answer your question sufficiently, or?
Yeah, thanks so much.
Oh, you're most welcome.
More questions? If you want to ask in Portuguese, you can also write in the chat and we can translate.
Yeah.
While people are thinking, I also have a couple of questions.
Sure.
So the first is, how do you motivate initial adoption for new users? Because the advantages of having this kind of relational database will become more apparent just as information accumulates. So how do you motivate users to migrate from other platforms or to start from scratch? So that's for new users, and also for people that are customers that may be using other platforms and they have to migrate a lot of historical information. How can you make that easy or at least doable for new adopters?
Yeah, number one, what we do is people who are wondering, I mean, we have blog articles. So on our blog, I have quite a number of articles that have been written about why would you consider this? So like, what's the advantage of having a relational system? In fact, I have one whole blog article just on that topic of a relational architecture. So what we usually do, we engage somebody, we say, send us some of your files, some of your nurseries, a few experiments, and that we build a Genovix database. Usually only takes like half an hour of our time or less. Then we show them what Genovix looks like with their data. And usually they say, wow, it's amazing what I can do. So as the philosophers once said, the proof of the pudding is in the eating, right? So they really need to see it, especially with their data. That's the most compelling thing. They start thinking about all the time that's saved. Importing, well, yeah, we spent probably some of our programs over a year having a lot of import algorithms. So you could, we actually have helped import breeding data from competitor software. Some people have looked at our system, said, you know, we really want your system. So we spent a lot of time on importing it and then linking the data. So what happens is if you have a germplasm file and you import hybrids with male and female parents, tell the system you're importing genotypes, here's the male female parent. And when you're done, the software automatically builds all the linkages. So you go and you click on one of the parents, it says, okay, this parent now is involved in 21 hybrids, and so far we've got 3,000 descendants. It automatically populates it up. So that's kind of what we've done in the interface. We spent a lot of time looking at it, making it fairly easy. Because like you say, the first thing is, it looks great, but can I ever get my data in the system, right?
Yes, exactly.
Yeah.
And the second question is about flexibility. So you've shown that users can write their own scripts to analyze the data. So I was wondering first if there's any kind of shareable knowledge base or something someplace where users can go and deposit their analysis scripts, or if they can share code. And also if, because you already have a pretty comprehensive list of designs, if it's possible to build upon those types of analysis, or if it's always from scratch that the users have to write their analysis.
Oh, you're talking about getting into R. Well, you do need to know some basics of R to make that work, like what model. So some of our scripts are there that you can actually go in and modify. So okay. What I showed was the big thing is setting up the data frame when it comes out of Genovix, and then capturing the means, typically the means and the adjusted values back in the system. That's the important thing. And in the middle, you will find, if you go to the R packages libraries, there are a lot of examples there already in the R community. So that's what we would kind of suggest. But beyond that, I think it's shared knowledge among people that know R.
Okay, thank you.
Yep.
So I think we have a question in the chat. Dr. Dieter, thank you for your talk. How does Genovix mobile work? Does it depend on a desktop version? Is it connected to a specific server, or can it be built upon a private server?
Oh, it connects only to Genovix. So here, if I go to controls and I go to remote data manager, so it starts in the database. So I create what are called packages. So here it'd be, actually we had USP experiments right here. There we go. So I've got this one here, and I can say I want to take data on these two traits. So at the moment, it's still in Genovix. So we've done it this way because some of the bigger companies, they, and some universities too, who are very productive of their data, they're contracting out to different people to get the data, you know, or maybe it's summer students that don't need to see the database. So what happens is this is then sent to somebody. So if I say create the package, the name is say USP 102 or whatever. And then I say here it's a pending package. Then here I'm going to send it to somebody. In this case, it'll be to myself. And then I'm going to click here, send. So at this point, it's sent. and that person is registered in the database, is known in the database. They need not be a user. So it actually, then when they open up their application, they click on connection here. They've got a bucket on Amazon where it's gone. So now it's sitting on the cloud. Okay. So now if I go to Genovix Mobile here, so I'm going to log in with the same username as in the database. And then if I go to manage packages, download new packages, with a bit of luck, I should see the USP one. There we are. I sent it just now. Okay, it's downloaded. I go to home and there I am. You see, and at this point, I can set up all my settings, timer, how fast, plot direction, all that. So I go to home and I say, okay, the grain yield, it's going to be 55. And I'm going to, I'll just put in 1 or 2 here, interest of time. And I'll say 66. I can look at the data so far. It's only this, whatever. So then when I'm done, I do manage packages again. I'm going to upload the current package. So now it's back to the Genovix database where it is hosted. It could be on the cloud. It could be on your server. It could be private cloud. But it does not just go to any place. It goes specifically to a Genovix database. So now I go back to Genovix. So I can go to receive packages. And there we are, USP. And I click here to process the package. And I guess I just saved 1 data point. Maybe that's it, not very much. And then I click here to apply. So we have this step so you can validate the data, that it is correct data. Because if you go all that work to have a really great database, it's kind of defeats the purpose to have data go in that's maybe not the best data, not correct or whatever. All right, so now I go to, it was USP ADT, I believe, or ADF. And there we are, grain yield 55. So there we are. So I hope that answers the question, like how does it work? I could take photos too, by the way, at the same time and upload them.
I'm guessing it also integrates with the QR code you showed earlier.
Yeah, that's right. So you could barcode something and then it will, it's also done by Bluetooth. So you have your Android and you can even have a Bluetooth scanner, scan something and it'll jump to that plot that it's found. And we're just finishing now the integration with balances, also by Bluetooth. So it's very flexible and very fast. Yes.
Thank you. I think we have time for one more question, perhaps.
Too much information.
Yes.
Okay, I have one more.
Okay, go ahead, Baldini.
Okay. How can we move forward for our university to have a license to teaching and research?
Well, just to talk to Henri, get the specifics, but we can give you a quotation. We have a special university price, which is a very big discount off the corporate price. And yeah, you just tell us what you want. We can give you a good quote and help you get started. Yeah.
Okay.
So, yeah.
I will contact you.
Okay.
Yeah. Thanks.
Yeah.
Okay, so if there are no more questions, I'd like to thank Dr. Dieter for the time and for the excellent presentation. Thank you very much for being with us. And also thanks Henry, I understand you work with our group to make this happen. So thank you for.
Yes, we'll do so.
Mm-hmm. Yeah, thank you.
For your time as well. And thank you all for being here with us. And see you
Okay.
next.
Okay.
Okay. Yeah.
Bye-bye.
Bye-bye.